Hello everyone, welcome back to BIOME, Biology, Tilse. Subscribe to BIOME channel for getting new notification about the new videos and don't forget to place the bell icon and while playing video modes, just check into the I button for getting information about the another videos. Today, I need to share something with you. Actually, इस चैनल में व्यू तो आ ही रहा है बट आप लोग सब्सक्रिप्शन नहीं करते. Please subscribe the BIOME channel. It helps us to grow. Your single click into the subscription button. It means too much for us. Okay, your single like, your single share, it means too much for us. Okay, so it's a kind request to all. Please subscribe the BIOME channel and try to subscribe it and try to share it among with your friends and peer groups. Okay, so today starts with the megasporogenesis and the embryo sac formation. Now, the events of the megasporogenesis I already discussed, but even though those students who missed the video, for them I'll definitely try to cover these events of the megasporogenesis for a while. And if you don't check the detailed process of the megasporogenesis, you can look back into the previous videos also for complete knowledge. And one more request to you, please don't skip the videos. Watch the entire videos till the end. Then only you can able to get the new concept. Okay, you'll able to get the total inter concept. Okay, skip करोगे तो कैसे पता चलेगा पूरा डिटेल कंप्लीट है. को नॉलेज लेना है तो पूरा वीडियो देखना है. ओके, चलो शुरू करते हैं. So the megasporogenesis. So what is the megasporogenesis? So the megasporogenesis means the formation of megaspore is called as megasporogenesis. Okay, तो मतलब क्या है? ये मेगास्पोर का जो फॉर्मेशन होता है इस प्रोसेस को हम लोग क्या बोलते हैं? मेगास्पोरोजेनेसिस. अब ये मेगास्पोरोजेनेसिस कहाँ पे होगा? Generally, the megasporogenesis took place inside the megasporangium. And the result of the megasporogenesis is the formation of megaspore. Formation of megaspore. अब ये megaspore कहाँ से बनेगा? The megaspores are generally formed from the megaspore mother cell. Generally, the ploidy of the megaspore mother cell is diploid. Now this diploid megaspore mother cell converted into megaspore. Okay, and what will be the ploidy of the megaspore? They are haploid. That means the megaspore mother cell they converted into megaspore, which one is haploid. But how it is possible? Over here through the process of meiosis. Okay, we know the meiosis is reductional division. करेक्ट. म्योसिस इज रिडक्शनल डिवीजन. सो यहाँ पे रिडक्शनल डिवीजन होगा तो डिप्लॉइड किस में कन्वर्ट हो जाएगा? हैप्लॉइड में. करेक्ट. अब यहाँ पे मेगास्पोर कितना मेगास्पोर बनेगा? टोटल यहाँ पे 4 मेगास्पोर बनेगा. As here there will formation of the fair megaspore, that's why here it can call it's a formation of megaspore tetrad. And 4 मेगास्पोर का जब फॉर्मेशन होगा तो उसके अंदर में 3 मेगास्पोर डिजेनरेट हो जाता है and 1 megaspore it will later further developed into embryo sac. इसका मतलब क्या है? 3 embryo sac जो है और 3 मेगास्पोर जो है ये डिजेनरेट हो जाता है. 3 मेगास्पोर क्या होता है? डिजेनरेट हो जाता है और 1 मेगास्पोर फर्दर embryo sac में कन्वर्ट होगा. करेक्ट. सो लेट्स लुक बैक टू इनटू दिस पिक्चर. इसके जरिए हम लोग जानने का कोशिश करेंगे कि यहाँ पे किस चीज का फॉर्मेशन कैसे होगा. So this is the entire megaspore mother cell. What I told, that is the entire megaspore mother cell, and this is the nucleus of the megaspore mother cell. Okay, that is the nucleus. Nucleus and nucellus actually both are not same thing. Now, if you come to the concept, so this cell is the megaspore mother cell. So the megaspore mother cell, we know they are diploid, and surrounding the megaspore mother cell here we are getting the presence of the nucleus cell. And what is the function of nucleus cell? Yes, you are correct. It helps to supply nutrition. It helps to supply nutrition to the megaspore mother cell. Now this diploid megaspore mother cell, it will undergo to the meiosis 1. And during the time of meiosis 1, first there will be karyokinesis. So due to the karyokinesis, two nucleus will form, and after that there will be cytokinesis. Due to that, between the two cells, the cell wall will form. That means from one cell there is a formation of two cells, 1 and 2. So that's why here we can call them as मेगास्पोर डायर. सो एक मेगास्पोर मदर सेल से क्या बना? मेगास्पोर बना. अब कितना मेगास्पोर बना? यहाँ पे दो मेगास्पोर बना. तो as there is a formation of two megaspore, that's why we can call them as megaspore dyad. Is it clear? Again, this megaspore dyad, it will be undergo to the meiosis 2. Here will be also first karyokinesis, means division of nucleus. After that there will be cytokinesis. So karyokinesis first होगा, तो यहाँ पे meiosis होगा, तो ये न्यूक्लियस जो है ये डिवाइड होके दो न्यूक्लियस बनाएगा. यहाँ पे दो न्यूक्लियस बनाया, ये न्यूक्लियस डिवाइड होके भी दो न्यूक्लियस बनाएगा. तो यहाँ पे टोटल 4 न्यूक्लियस का फॉर्मेशन होगा. देखो इसमें टोटल 4 न्यूक्लियस है. अपर 1 2, लोअर 1 2. एंड कैरियोकाइनेसिस के कारण में यहाँ पे 4 न्यूक्लियस बना, but due to the cytokinesis in between them there is a formation of the cell wall. Now how many megaspores are formed ultimately? There is the formation of total 4 megaspore. 4 megaspore. So as over here there is a formation of 4 megaspore, that's why here we can call them as megaspore tetrad. And once the megaspore tetrad will form, so out of this 4 megaspore, the 3 megaspore towards the micropylar end, what I told, 3 megaspore towards the micropylar end, they will degenerate. Okay, इसका मतलब 4 के अंदर में जो 3 मेगास्पोर है towards the micropylar end, ये लोग degenerate हो जाएगा. And the megaspore towards the chalazal end, it will be further developed into embryo sac. It will be further developed into embryo sac. Clear? So next we'll come to the embryo sac formation. Now if we come to the embryo sac formation, now for the formation of embryo sac, total how many megaspore is involved? Okay, so we already discussed that 3 megaspore degenerated and 1 megaspore ultimately will be developed into embryo sac. So ultimately from 1 megaspore only the embryo sac will form. So that's why such type of development is called as monosporic development. So suppose over here the 2 megaspore formed helps in the formation of the embryo sac. That conditions we can write as a bisporic condition. Okay, bisporic condition. Suppose the 4 megaspores are helped in the formation of embryo sac. That will be the tetrasporic condition. Okay, so the details of this monosporic, bisporic and the tetrasporic development of the embryo sac already I discussed. If you're interested, you can look back into the previous videos. Next comes to the during the formation of the embryo sac, what are the changes will take place? First, what will happen? The single nucleus is left. That single nucleus, it will be undergo to the mitotic division. So yes, we are correct. Mitotic means it's an equational division, it's an equal division. So due to that one, the nuclear number will increase. So one will form the 2 nuclei. Again the 2 nuclei, again they will divide, they will form 4 nuclei. Okay, they will form 4 nuclei. Again the 4 will divide mitotically and again they will form 2 nuclei. So ultimately how many nuclei will form? Ultimately there will be formation of total 8 nuclei. And once the 8 nuclei will form, then there will be partitioning of the 8 nuclei, and as a result there will be formation of entire complete embryo sac. Okay, so let's look back into some previous picture, and through the picture we'll try to discuss this. So this picture already I told. So here it's divided, megaspore diad is formed, and ultimately megaspore tetrad is formed, and these three are already degenerated and only this one is left. And this one again it will be going to divide mitotically. So the mitotically it means whatever, simple things, it's a by the process of mitosis. So mitosis, we know it's an equational division. So due to that one, what will happen over here? If the single cell is going to divide mitotically, so as a result, there will be formation of total two cell. So as the two nuclei will form. So the two nuclei again, it will be comes to the mitosis process. Mitosis. So during this mitosis process, here only the karyokinesis will be there. During the karyokinesis, what will happen? Total two nucleus is formed, and again this two will going to divide by the process of mitosis. This one it will form again two nuclei. This one again it will form the two nuclei. Correct? And again, if it is going to divide, so as a result, what will happen? So there will be again mitosis. Due to the mitosis, what will happen? These two again will divide, these two again will divide. So as a result, there is a formation of total four, and these two again it will, if they will divide, so this will form two, this will form two. So again this set it will be four. So total 8 nuclei will form. And once the 8 nuclei is formed, that means that by the process of karyokinesis, still now the cytokinesis not took place. Later what will happen? There will be cytokinesis, and due to the cytokinesis there will be a partitioning between the nucleus due to the formation of सेल वॉल. इसके मतलब क्या है? सारा न्यूक्लियस के अंदर में क्या होगा? सेल वॉल का फॉर्मेशन होगा. अब जब से सेल वॉल का फॉर्मेशन हो जाएगा, तब धीरे धीरे क्या हो जाएगा? एज द सेल वॉल इज फॉर्म्ड, तो सारा न्यूक्लियस के अंदर में सेल वॉल का फॉर्मेशन हो जाएगा, तो एक एक सेल बना लेगा ये लोग. So just look back into this picture. Just assume these three upper cells, okay? These three upper cells, they are present towards the chalazal end, and in between this nucleus, among this nucleus, the cell wall also formed. So here this is one cell, here this is one cell, here this is one cell. So this is, we can call them as antibodal cells. So where antibodal cells are formed? Close towards the chalazal end. Just look back to into these two nucleus. These two nucleus, suppose here one, and in between them also the cell wall is present. Correct? So that's why this is a one cell also, this is one cell also. Just take now this one. So this one came here, and they also formed a cell wall, and we can also call them as egg. And now this one is left and this one is left. This both of them, it will come towards the center region. Look back to here, and they will form a two nuclei. And in between, around them, there is no formation of cell wall. So that's why we can also consider them as a single cell. Correct? Now if you calculate total how many nucleus is present, so here just calculate the total number of nucleus. So the total number of the nucleus, it will be 1, 2, 3, 4, 5, 6, 7, 8. So total 8 nuclei is present. But if you check how many total cells are present, if you check total how many cells are present, so that's why, let me erase this. So total number of cell. So now if you come to the total number of cell, so this is a one cell because it's partitioned to the second cell, third cell, फोर्थ सेल, फिफ्थ सेल, सिक्स्थ सेल, तो 6 सेल तो हो ही गया. बट देखो ये दोनों के बीच में कोई सेल वॉल नहीं है, दोनों बीच में दोनों कोई सेल वॉल नहीं है, but that's why we'll consider this one as a single cell. That's why we'll consider this one as a single cell. So now 1, 2, 3, 4, 5, 6, and if you consider this as a single cell, that means the 7th cell. That means inside the mature embryo sac, that the formation of total 7 cell and 8 nuclei. 7 cell and 8 nuclei. Okay. So just look into this picture. So here it's written The mature embryo sac is 7-celled and 8 nucleated condition. Okay, clear? So how it is formed, we already know. Total, count the total number. So there are the total 2 synergids. Remind the synergids always finds close towards the chalazal end. Close towards the close towards the micropilar end. Okay, micropilar end. And this end is the chalazal end. So 2 synergids, 1 egg cell, okay, then one polar or one central cell and 3 antipodal cell. So if count, so 3 plus 1 4, 4 plus 1 5, 5 plus 2 7. So that means the 7 cell. And if you count the total nucleus number, so antipodal cells, we know they have total how many nucleus? They have total 3 nucleus. Then the synergids, how many they have total nucleus? The synergies nucleus number is 2. In the egg cell, the nucleus number is 1, and in the central cell, the polar nuclei number is 2. Now if we count, so we will get total 2 plus 1 3, 3 plus 2 5, 5 plus 3 8. So that means inside the mature embryo sac, there are the total 7 cell and 8 nuclei condition. So just remind these things very nicely. This is very, very, very important. Okay. So thank you for watching Biome, and don't forget to subscribe the Biome channel and just click on the bell icon, and in between also videos you can click on the I button, and don't forget to share the videos and just comment and like the videos. Till then, thank you and goodbye. And very soon we'll meet with another new video. Thank you.